Well, this is a rather grandiose title for an eclectic collection of research projects that I've been involved in, some centrally, some only peripherally, over the last 2 years. And what I'm going to show you is some of the projects that we've been doing and where we've been doing them and why I think they're interesting in terms of being able to tell things about our continent. Up in the, for those of you who are interested, up in the top right-hand corner here is going to be a small-scale version of this map with stars on it. That'll show you exactly where the research was conducted. And for those of you also interested in such things, the references or publications that arise from this research will be down the bottom here. So, after such a grandiose title, and I'm not sure that I'm going to be able to actually live up to the nature of this title, I'm going to start with something a little bit corny, because Australia is a diverse and complex country with unique, diverse and complex organisms. And so the corny bit is I'm going to start with Dorothea McKellar. Okay, I love a sunburnt country, a land of sweeping plains, of ragged mountain ranges, droughts and flooding rains. I love her far horizons, I loved her jewel seas, her beauty and her splendour, the wide brown land for me. So what we're going to do here is explore this wide brown land. Most of the photos you see will have been taken by staff members or by people who are actually involved in the research, as all of those were. And so you're actually looking at real things happening in real time here. The first project that I want to talk about, and I'm going to outline 5, actually is in this landscape. And it's about conserving endangered lizards and what they can do for us. The leader of the research here is Adam Stowe, doing his bush tucker man thing, and Siobhan Dennison is one of the people working on this project. She's in the audience. The project is about this animal here. It's the great desert skink, also called the jaccara or the warrener, and it lives in family groups that share a burrow complex. In other words, mum and dad live in a burrow and they raise their teenagers and their babies in the burrow. That's very unusual for lizards. And Adam's interest is in genetics, ecology and behaviour of this organism across the Red Centre from Uluru out to Docker River near Western Australia and up into the Tanami. And the first thing that people usually say when I tell them that I'm going with Adam for 3 weeks into the desert to catch lizards is, why would you want to do that? Well, the first thing is because they're really cute. I really like these lizards, and I also really like the outback. Those of you who've never been to the Red Centre, you really should go there. I know I'm on tape here, but the Red Centre, the outback, is much more of Australia than Bondi and the Opera House ever will be, and you really need to go and see it. So here's the reasons, the scientific reasons why you might do this. Firstly, it's vulnerable or endangered across its entire range. It's of great cultural significance to the local Aborigines, and the local Aborigines are always very, very happy when they see a jaccara, because these things were thought to be extinct in South Australia and were only rediscovered very recently. It can help us identify what are called refugia from past climate changes, and I'm going to tell you what those are in a minute. And it can tell us about the evolution of social behaviour and parental care. I'll just explain a little bit about this. This lizard has closely related lizards, different species, that live in monogamous pairs but don't raise families with them. There are also closely related species that are solitary as well. And so within this one group of lizards, we've got the entire progression from being, you know, your nasty typical lizard that lives by itself and does nothing for anyone else, to being monogamous, to actually living in family groups. So one of the exciting things we're thinking about doing and investigating at the moment is trying to identify the genes that are responsible for that social behaviour. There are clues from the Human Genome Project and from work on prairie voles, and we're going to try and apply that to understanding social behaviour and parental care as it's happening in this particular lizard group. So what do I mean? Let's go back and talk about identifying refugia from past climate changes. So we can use genetics, we can use analysis of lizard diversity to actually tell us things about what's happened in the past and about not what's happened to the climate, but what's happened to the organisms that lived in those climates. So past climate changes have changed the distribution of plants and animals. 21,000 years ago, you can see that the Indonesian archipelago was very different in vegetation to what it is now. You can see that the centre and margins of Australia were very different. Imagine that over very broad timescales, climate changes, what happens is species contract to small areas called refugia. When climate gets a bit better, they expand again. That means that the places where species contract to are actually exhibit genetic diversity in that species. So if we can find genetic diversity in the distribution of a species, that will correspond to past refugia, and they are the places that should be conserved. And so where Jacare and other species exhibit increased diversity, that's where the refugia from past climate changes have been, and they are the places that we need to identify as conservation priorities for the future, because we now face even faster and more dramatic climate change than has happened in the past. And you can see, you know, these guys are diverse. It so happens that one of our prime study sites, Newhaven, is run by the Australian Wildlife Conservancy, and that is a hotspot for genetic diversity of Jacare and presumably lots of other organisms as well. So that's good news. We can also use genetics for estimating dispersal and future distribution of organisms. And this is work by Ted Duckett, one of Adam's PhD students. He works on this little cute fellow here. This is the tree detella, or it's an arid zone gecko. And by looking at genetic diversity in this species and looking across its entire distribution, which is here, he can predict how fast these lizards move over time. He can say, look, they move maybe about 8 kilometres a year under these circumstances. We can take those data and we can marry that to projected distributions in the future. And that means that we can track where species are likely to be in the future, and we can make sure that they've got a way of getting there. The best example here is look at this distribution here. Here's the gecko at the moment. In another 20 years, this is going to be its distribution. This is where it's going to want to live. And by 2070, this is where it's going to be. So how are the geckos that are here going to get down through here and to here over that time? Well, clearly, we have to leave corridors open so that they can make it that way. These are the kinds of things we need to think about if we are going to help conserve species over the next 100 to 500 years of climate change. Okay, let's now talk instead about something that we don't like. You all know that the rabbit and the cane toad were disasters being introduced into Australia. And it's interesting because, you know, often these animals and plants are introduced for the best of motives, and yet they get out of hand really quickly. They create havoc in Australian ecosystems, but we don't really have an idea of why some species can spread quickly and others can't. So how have exotic species been introduced and spread? Can we reconstruct their history? Now, many of you will know about this little guy here. Whoops. This is the eastern mosquito fish. It's the most invasive freshwater fish in the world. It was introduced around the world to control mosquitoes in the 1920s through 1940s. And of course, it ate everything except mosquitoes. Sounds like a pretty typical story, like the cane toad. It's pretty nasty in Australian freshwater ecosystems. It eats everything and you know, basically kills native fish. What we wanted to do, and you can see our collection locations here, mainly from collaborators. We didn't go to Western Australia to collect these guys. Understanding past introductions may help us prevent new ones and understand what's going to happen to new introductions. So this work, the DNA work in this case, was mainly done by Hilal and Yagiz, who are also in the audience tonight. What we did, we collected fish in the field, we tested them using DNA analysis, and we used that DNA analysis to reconstruct the history of their introduction and spread, because the stories that go with this fish are actually not very clear. The original story is that the fish comes from the US into the Iberian Peninsula, Portugal and Spain, from there to Italy, and in 1922 from Italy to Australia. We tested that hypothesis and it turns out it's not true at all. DNA analysis does help. The colours here just represent different kinds of a particular DNA marker that we use, and the marker that's common in Italy is this red one here. So clearly, you know, it's possible that stuff did come from Italy. But there are also some markers. The blue one, for instance, has only ever been described from France and Greece, and that green one there, which is just present just at this one location on the south coast, that's only ever been found previously in the Potomac. So clearly, there are multiple introductions here, introduced from both Europe and the USA, and there's been movement by humans, because how else does a fish get over here or to Tasmania? What we're doing now is using finer scale markers to ask particular questions about movement between catchments. So for instance, it's possible that movement across into the back of Queensland has then led to the fish being able to run down these catchments and colonise around Lake Eyre and perhaps even down into the peninsula here. But we can answer that using better markers to track more precisely the movement of these fish. Now I'm going to talk about something to do with humans. You know that antibiotic resistance in bacterial pathogens is a huge problem for in medical circles. And a lot of the work that Hatch Stokes from UTS and myself have been doing over the last probably 5 years has to do with tracking where these antibiotic resistance genes come from. And it's pretty obvious they come from the general environment, into bacteria that are pathogens of humans or that live in or on humans. Now, we've got a situation where genes and the bacteria that contain them can basically move from any of these spheres directly from humans to their domestic animals, to their companion animals, to wild animals, back to the environment. And what we're now interested in is the second order effects of antibiotic use, and that is what is happening to the resistance genes that are in humans. So every single person in this audience has antibiotic resistance genes in the bacteria that live inside their digestive system. Guess where those resistance genes are going? They're going down pipes into the ocean and sewage treatment plants and etc. I'm interested in what's happening to those genes. So here's an example, and this is work done with Michelle Power. This cute little fellow here, this is an endangered rock wallaby, and because natural populations of this rock wallaby are declining, we breed them in captivity and then release them into the wild. Michelle and I have discovered that the captive bred rock wallabies have actually picked up antibiotic resistance genes that are typical of human clinical isolates. You know, we can DNA sequence them and we can tell what's going on. So by having them in close proximity to human activities, they're exposed to human waste streams. That means they pick up these genes, and then of course they release them into the wild. When we look at wild rock wallabies, we find no antibiotic resistance genes at all. So we're interested in tracking where these genes are getting and how far they're getting. And this really cute photo here is one of the released animals wearing a radio collar, shaking hands with one of the wild animals. Pleased to meet you, have a few antibiotic resistance genes. There is also potential problems for humans as well. So resistance genes spread from humans into bacteria that are found in the digestive system of king prawns. So if you take eastern king prawns from anywhere off the coast and you look at the, you devein king prawns, you should devein king prawns, and this is why. Inside those veins, which are really the digestive system, is an organism called Acinetobacter, which is an emerging opportunistic pathogen in hospitals. What we've discovered is that that particular species, and there's a group of them inside the king prawn, has picked up antibiotic resistance genes from human sources. It has then supplemented those resistance genes with some extra genes that confer survival under animal immune defences. And so those bacteria that have come from humans have donated antibiotic resistance genes to bacteria that live in prawns. We eat prawns. We often don't cook them very much. We often don't devein them properly. So there's a clear passage straight back into humans for an opportunistic pathogen that is not only antibiotic resistant but contains other genes that are going to make it more pathogenic. And we are predicting that that is going to become a disease problem for humans. We've discovered it before it's appeared in hospitals, but it will appear in hospitals soon enough. Okay, so now I want to talk a little bit about managing vegetation, because we now have 7 billion people on the planet, and humans are the largest users of fresh water, and freshwater usage per capita has been declining for a long time. In fact, access to water, you only have to listen to the news. Yesterday, we were talking about environmental flows in the Murray-Darling. Access to water and the amount of water that's available for human use is going to lead to conflict in the future, not just between states, but between countries. So we are actually using much more water resources than we need to, and in fact we're placing now great strains on groundwater by extracting it. So up on the north coast of New South Wales, there's a lot of groundwater being extracted, and we want to be able to manage that use and understand what effect the water use will have on above ground vegetation. So this is a kind of a very practical project that we did with the New South Wales Office of Water. Underground ecosystems are really difficult to investigate. If you dig down into the soil and you find a root down there, you don't know what it belongs to. You can't trace the root back to the plant. So what we did was, with Steve Griffith, who's the other person who worked on this, along with Hillel Varinli, he took cores down to the water table, collected roots at the water table. We took those roots, DNA sequenced chloroplast DNA out of there, and matched it with material that we collected from the surface. And we were able to say, look, these particular plants of this particular assemblage, which is called Wallum, are dependent on groundwater. And we anticipate that anything that is groundwater dependent can be a sentinel for excessive water use. We know this because excessive water abstraction in Western Australia has led to the decline of a whole series of vegetation assemblages in Western Australia. And we had some surprising results. Large trees like eucalypts and banksias obviously have deep roots, but so also did a number of small perennials like these guys, just little shrubby things and sedges. You know, they're only about this high, but their roots can go down 2 or 3 metres. These are the kinds of plants who are most going to be affected by excessive abstraction of groundwater. So once people do start to remove too much groundwater, you'll see it by these guys dying, long before the banksias and the eucalypts show problems. So here's a DNA method of just managing that groundwater use. Okay, for the last project, I'm going to stay underground. I'm going to talk to you about aliens under the Nullarbor. So you all know the Nullarbor Plain. What you might not know is that it's the largest slab of limestone in the world. That's why it's dry and flat and has no trees, and most people again would say, well, you know, what would you want to do on the Nullarbor? Well, it hides an underground secret, because where you've got limestone, you've got caves, and if you can find these caves, below the desert there are caves full of water, about 60 metres below the surface, and this is slightly salty water. So imagine you're travelling across the Nullarbor and you happen to bring your abseiling gear and you also happen to bring your scuba gear. You could go for a bit of a scuba in one of these. And here we go. You have to be seriously insane to do this. This is the most dangerous recreational sport on the planet. The reason for that is that above those divers, that's rock. That's not the surface. If your air runs out, there's nowhere to go, because these are completely water-filled tunnels, and it takes, to get to the end of the tunnels as far as you can go, it takes 16 hours return journey. So that's a pretty serious undertaking. However, I'm pretty glad they do this. There's no way you'd get me doing this. But have a look at the, well, firstly, have a look at the water clarity in these caves. It's amazing. But have a look at the wall here, and you can see all this fuzzy stuff. See all that hairy stuff? If you look closely at that, it looks like this. These are called microbial mantles, and this is a purely microbial ecosystem. It's just bacteria and archaea. In other words, not complicated cells, just simple prokaryotic cells. And it thrives in the total darkness of the caves, and all of the components of this ecosystem are undescribed species. 20 to 50%, depending on where you draw the line, are actually undescribed phyla, for those of you who are interested in that kind of taxonomic ranking. We have been working on this to try and find out what it is, what components make up this ecosystem, and how it works. And with Ian Paulson from CBMS, we've recently started doing what's called metagenomic DNA sequencing, which means we just take everything in the cave and DNA sequence and reconstruct the genomes of the organisms that are in that cave from scratch. And doing that, we found that this is how this ecosystem works. Now, normally, on the surface, and most biology textbooks will tell you that Mr. Sun shines down, plants gather solar energy and convert that into chemical energy, which is used to make glucose, which feeds everything else on the planet. This ecosystem is in permanent darkness all the time, completely separate from the surface, completely separate from photosynthesis, and it makes its living this way. It takes ammonia that's present in the water. Ammonia is the stuff that you smell in babies' nappies or cleaning products, and both archaea and bacteria oxidize this through this pathway here to nitrite. That generates energy in the form of ATP, for those of you who are interested in that kind of stuff. And then that nitrite is oxidized to nitrate, again generating energy from what's called chemosynthesis instead of photosynthesis. This ecosystem is probably present completely under the nullarbor. The whole nullarbor plain underneath probably has ecosystems like this. So this is a huge chunk of Australia with an alien ecosystem completely independent of the surface and completely independent of photosynthesis. It's a pretty amazing thing. What we are aiming to do now is to understand every single organism that's in this ecosystem and understand every single piece of biochemistry that drives it, so that we can, for the very first time, completely describe an ecosystem from the gene level up. That's a, will be, if we can do it, will be a world first. Okay, so that's where I want to end. I'd just like to finish by saying Australia provides just endless opportunities for research, because our animals, our plants, our landscapes are all unique. And there's still lots and lots to discover, amazing things to discover, such that, you know, I wish there were 5 or 6 of me so that I could do all the stuff that I really want to do. And if there are any, I'll now take some questions. Thank you.